Well, hello everyone, my name is Charles Tyler and I'm based at the University of Exeter. I'm going to describe to you today some of the work we've done under Work Package 5 focusing on using the zebrafish model. I'll describe three pieces of work that we've done collectively. The first is using bioinformatic tools to help us better understand the gene regulatory networks and response pathways which are common between that of the zebrafish and mammalian systems, including humans, trying to develop this so-called biological read across. The second component is looking at the application of a transgenic fish model to help us better establish where zinc oxide and silver nanoparticles interact in the bodies of fish to mediate biological effects. And the third piece of work is work conducted by colleagues at Heriot-Watt University looking at time-related expression of inflammatory genes in the respiratory system, i.e. the gills of the zebrafish, for exposure to titanium dioxide nanomaterials and how that relates to what we know in mammalian systems. And the focus throughout this work has really been on oxidative stress and inflammatory mechanisms, and we're trying to better understand the commonalities in responses between the zebrafish and mammals, including humans. I probably don't need to introduce too much with respect to the zebrafish as our study model. It's clearly widely adopted in the OECD test guideline studies. It's used widely as a model for developmental biology, has been for some time. You can visualize the whole process of embryogenesis in a period of 96 hours through these translucent, transparent eggs. It's widely used in medicine for understanding human diseases, particularly developmental diseases, and for their treatment too. Importantly for patrols for this part of the project, for both the informatics work and also the transgenic fish work, it has a sequence and well annotated genome. And it represents, in terms of thinking about protection of the natural environment, it represents one of the largest families of freshwater fish globally. So in terms of the informatics works we've done basically here, we're looking to try and better establish understanding in terms of gene regulatory networks and gene response pathways for the effects of engineered nanomaterials between zebrafish and mammals to better establish to what extent we can read across between our experimental model of the zebrafish and that occurs in mammalian systems. And here we adopted two approaches, a so-called targeted approach and then an unbiased sort of genome-wide approach. In a targeted approach, we surveyed all the available literature and identified all the possible sort of adverse effects which have been reported in fish, and they included things like through oxidative stress, inflammation, respiratory distress, neurological damage, and defects in embryo development. And many of those effects have also been reported in mammalian systems too. Oxidative stress was one of the most commonly reported responses, and here we're able to identify 17 genes which were activated by metal-based nanomaterials, and all of those genes too were common to those which have been reported in mammalian systems. And so certainly for oxidative stress and inflammatory response pathways, it looked like to be strong commonalities between the zebrafish and that in mammalian systems. We then look to use genome-wide approaches to try and develop a depth of understanding and confidence in terms of those pathways, particularly oxidative and inflammatory response pathways, in terms of the representation of the genes actually that map from the zebrafish onto those mammalian response pathways. And so we look for so-called zebrafish orthologs within the sequence genomes for the zebrafish. We also looked a little bit in terms of some of the other fish species too. We identified a whole series of orthologs that actually mapped onto both the oxidative and inflammatory response pathways which have been established in mammals. We then looked to see what genes were actually expressed in relation to the mammalian system for exposure to nanomaterials in different tissue types, and set out to identify how many zebrafish orthologs we could map against those expressed genes for mammals across those specific pathways. And for oxidative stress, we were able to find over 100 zebrafish gene orthologs which were expressed for the liver. We then look to map those orthologs onto so-called KEGG pathways. KEGG pathways are pathways that provide all the information in terms of the genes involved, in terms of regulatory networks and response pathways for mammals. And here we're just looking for part of the oxidative stress response pathway for glutathione metabolism. And the green boxes indicate where we are able to identify zebrafish orthologs mapping to components of that glutathione metabolism response pathway. And we were able to show high representation of genes onto that pathway, zebrafish orthologs onto that mammalian pathway. And this was true also for other components of the oxidative response pathway, and also for components of the inflammatory response pathways too. So these findings strongly support the use of the zebrafish for helping us understand the regulatory and functional networks associated with oxidative stress in mammals. And so therefore the potential also for the use and application of the zebrafish for predicting outcome for exposure to engineered nanoparticles through these response pathways in mammals. The second component of work we did was looking at the application of a transgenic zebrafish model to look at the susceptibility of them to the effects of zinc oxide and silver nanomaterials. And this oxidative stress model is a transgenic we've created, works through something called the electrophile response element. The electrophile response element is a sort of master regulator of the oxidative response pathway both in mammals and in fish. And what we did, we stably integrated 3 electrophile response elements into the genome of the zebrafish, and our transgenic zebrafish are linked to a fluorophore M cherry, such that if you get activation of that electrophile response element, you get this red fluorophore M cherry being produced, and you can visualize that response then actually across the whole organism. In fact, you can visualize it in real time too. And we've already used this zebrafish for oxidative stress to study a whole variety of different sort of chemical stresses, being able to identify tissue-specific responses to different types of chemicals stresses, and in the zebrafish, and we've been able to show they've mapped rather nicely to what's known in mammalian systems. In this work under patrols, we applied this transgenic zebrafish to was able to identify which is highly responsive for exposure to zinc oxide nanoparticle was the olfactory system, the olfactory epithelium, the olfactory bulb. And when she exposed the zebrafish throughout development, so from fertilization through to 96 hours post-fertilization, she was able to show that exposure to zinc oxide nanoparticle caused disruption in the development of the olfactory epithelium and olfactory bulb system. And so the top left panel is showing the responses in the olfactory system to exposure to zinc oxide nanoparticles, and the cartoon is showing what elements of those sort of neural systems are affected in terms of their development, what they look like as a consequence of that exposure. The bottom left-hand panel actually shows you the responses that occur in those fish for exposure to zinc oxide nanoparticles with respect to within a double transgenic fish. So here the transgenic for oxidative stress has been crossed with one where you can visualize the macrophages within the zebrafish, and the very bottom left hand of that panel shows you, the green spot shows you the macrophage migrating to the olfactory system, which has been exposed to zinc oxide nanoparticles. So those exposures to zinc oxide nanoparticles, which are causing this disruption in terms of development, are also inducing this sort of inflammatory response. I then actually took those larvae, which you exposed during early life, and she did some behavioural tests, and she was able to show that the ability to detect a stress odor was impaired as a consequence of that early life stage exposure. So the larvae were placed in these chambers and allowed to move freely within these chambers, and then stress odorants were introduced into the chambers at both ends of that chamber, that exposure chamber, and then a movement was mapped. The movement of the exposed versus non-exposed fish were sort of mapped within that chamber, and she was able to show that they were less responsive, less able to respond to the stress odors, those which have been exposed to zinc oxide nanoparticles. So here's a very good example of an exposure during early life has consequences chronically in terms of subsequent health and fitness of the zebrafish. The last piece of work I'll describe is the work that was carried out by Heriot-Watt University, and here they're looking at time-related expression of inflammatory genes in the gills for exposure to titanium dioxide nanoparticles. And they used a system where chambers that they developed whereby they can maintain aqueous exposure dispersions of these nanomaterials. They exposed their zebrafish to titanium dioxide at various concentrations, and then they measured gene expression at time intervals to look at the ontogeny expression of a whole series of genes which were involved in sort of the inflammatory response pathways. Those genes included things like chemokines and cytokines, and those which are involved in sort of mucus and mucus production. They looked at expression of those genes in one of the gills, and then they dissected the other gills out to do some histopathology to look at the sort of cellular level effects. In terms of the gene expression, just looking at 2 of the genes, they looked at interleukin 8 and here muco5AC. You can clearly see a nice sort of like a temporal response in relation to interleukin 8, as you would imagine, as is known to occur for interleukin 8 for exposure to this and other metal-based nanomaterials. And for muco5AC, very interestingly, there's sort of a suppression in expression of that gene. And these are responses which really typify and really mimic what is known to occur in mammalian systems. They've also looked and worked with colleagues at Swansea University through work package 3 to establish to what extent the responses they saw in the gills are mimicked in an in vitro culture system, which is a human cell culture system looking at human epithelial and macrophages. And certainly for interleukin-8, there appear to be some commonalities in terms of patterns, although there's somewhat delayed in terms of some difference in terms of the temporal ontogeny of that expression. For other targets, such as interleukin-1b, they didn't find that to be the case. But there's a whole series of other genes which they have yet to investigate. So just finishing up now in terms of some just key take-homes from the work I presented. First thing to emphasize is the principal pathways of these engineered nanoparticles through oxidative stress and inflammation appear to be highly mechanistically conserved from an evolutionary context between fish and mammals. And so it gives us good faith, confidence in terms of being able to read across from a biological context from fish to mammals for those effect mechanisms and pathways. The second key take-home, I think, really is that certainly iron regulation, olfaction are highly sensitive to selective metal-based nanomaterials, and for us at least it illustrates the need to better focus or stronger focus on the effects of some of these metal particles on sensory systems, not just in fish but perhaps in other organisms too. I was be able to show us through some elegant cell biology to exposure to zebrafish during early life stages to zinc oxide nanoparticles certainly disrupts the olfactory sensory neuron sort of developmental system, and that then impedes their ability then to pick up olfactions, certainly those in relation to sort of like stress related odors in later life. So a classic example of whereby early life stage exposure to these materials can have subsequent chronic effects for the health of those fish. And lastly, through the work of Harriet Watt and colleagues, they were able to show that a number of the genes that we know to be responsive for inflammatory response in the lungs and of human systems are similarly responses in fish gills. And but to what extent, so the in vitro system for those mammalian cell cultures can be used to compare and contrast with that occurring sort of zebrafish as is still ongoing work. Finally, just thank you for listening, and in particular I want to acknowledge Martina for her leadership of Work Package 5 and all the Work Package 5 team. Shireen, of course, in terms of leading patrols, done a fantastic job leading patrols consortium overall. And I've realised I've rattled through lots of pieces of science today, and if anyone have any questions, those are my contact deals. And thank you for listening.